Hi, welcome to today's OBSSR Director's Webinar. The title of the talk today is Social and Population Health Science Approaches to Understanding the Human Gut Microbiome. I'm Christine Hunter, the Deputy Director of the Office of Behavioral and Social Sciences Research at the National Institutes of Health. Before I introduce today's speaker, I have a few housekeeping items to mention. First, today's webinar is being recorded, and the recording will be available in about 2 weeks on the OBSSR website. Today's presentation will be followed by a question and answer session. All phone lines will be muted during the webinar, and questions and comments won't be taken via the phone, but will be taken using the WebEx question and answer or chat features. So if you want to ask a question or send a comment, click on Q&A, select all panelists, type in your question, and send. So feel free to send questions anytime during the webinar. And after the presentation, OBSSR's Dr. Mike Spittle will facilitate the question and answer session and ask your questions to the presenter. So I'm really pleased to introduce today's presenter, Dr. Pamela Hurd. Dr. Pamela Hurd's research expertise focuses on inequality and how it intersects with health, aging, and policy. She's an expert in survey research and biodemographic methods. She is currently the chair of the Board of Overseers for the General Social Survey, a member of the Board of Overseers for the Panel Study of Income Dynamics, a board member for the Population Association of American...
A board member.
Okay, and a standing member of a National Health Institute's review panel for the social and population sciences. She's received grant awards for her work from the NIH, the Russell Sage Foundation, the Ford Foundation, and AARP, and she's the principal investigator of the Wisconsin Longitudinal Study. It is my pleasure to welcome Dr. Hurd to present today on social and population health science approaches to understanding the human gut microbiome.
Hi, thanks so much for having me. I'm really happy to be doing this. For those of you listening out there, I'm currently on the faculty at Georgetown University, but I'm actually visiting for the year at the University of Oxford, the University of Oxford, specifically Nuffield College. So I'm speaking to you from the UK. And today, I'm going to talk about linkages basically between the population health sciences and research on the gut microbiome. But before I do that, let me just start by thanking some folks, some really important folks for supporting this broader body of work, and that especially includes the National Institute on Aging, and particularly the Office of Behavioral and Social Science Research, the division, as well as support from UW-Madison, who also helped support this project. I also want to specifically give a shout out to my collaborators on this body of work that I'm going to be talking about. A critical collaborator includes Federico Ray, who's in the UW-Madison bacteriology department. I want to say more broadly, you can kind of see on the slides there, we have a very large interdisciplinary team of folks who've been involved in some of the work that I'm going to present on. And I want to emphasize that that's really critical in the sense that I am a social scientist, a population health scientist, and I represent that part of this project. But of course, my collaborators are trained in the biological sciences as well as biostatistics. Let me just sort of walk through an outline for the talk to give you a sense about what I'll be covering and where I'm going. So I'm going to start really basic with what's the microbiome. So just to preface all of this, some of what I'm going to talk about today may sound, if there are biologists in the audience, it may sound a little bit basic to you, and if there's social scientists today, some of what I'll talk about might sound a little basic to you too. But I'm trying to cover the kind of broader swath of this work, and the collaborative nature of it implies that we're coming from very different bodies of work, and most of us aren't as familiar with the other discipline's body of work. So I'm trying to kind of manage that basically. So first I'm going to talk about what's the microbiome. Then I'm going to sort of make a case for why a social scientist or a population health scientist is talking to you about it. I'm going to talk a little bit then about what population health scientists have to offer to research on the microbiome and especially the gut microbiome, and I'm going to talk about this in terms of substantive contributions, contributions in regards to data, and also just broadly methodological contributions. And then finally, I'm going to finish off with a very specific empirical example using the Wisconsin longitudinal study, excuse me, that focuses on social relationships in the gut microbiome. What I'm hoping to do with this example is, in essence, to flesh out in a very particular empirical example what I've tried to lay out before in terms of what these kind of collaborations can offer in terms of what we can learn about the gut microbiome and specifically about the social determinants of the gut microbiome. So what is the gut microbiome? Or I'm sorry, what is the microbiome? So the slide I have up here is the cover of a Nature magazine from a few years back, where they're essentially talking about the microbiome, the human microbiome, as our other genome. And what they mean by that is the idea and the kind of growing body of evidence that might indicate that the microbiome is just as, potentially even more important in terms of shaping human health as the human genome. Now, I'm making a very kind of strong statement there. I don't mean it quite as strongly as I'm saying it. The work on the microbiome is a bit farther behind work on the human genome. But the body of work has been really rapidly accelerating, and there have definitely been some, I think, really important and interesting findings on its relationship with health. So in terms of the microbiome, though, we're an amalgamation of cells, both human and microbial, with a number of microbial cells largely exceeding our Homo sapien cells. So the these collection of microbes that live on us and within us are the human microbiota, or the associated genes, the human microbiome. Okay, so these are all of these microbes that sort of live on us and within us. There's trillions of them, and they are assumed at this point, based on existing empirical evidence, that they are shaping health. Now, I'm going to get a bit more specific here by focusing on the gut microbiome. So I think it is still the case that the largest body of work is focused on the gut microbiome. For the social scientists out there, and especially any demographers, this is just to try to give you a sense about scale. So if there are 7.1 billion humans on Earth, there are well over 100 trillion microbes in your gut. And these microbes have some important influences. So the gut microbiome is essentially considered a metabolic organ, It shapes features of our immune system. It processes nutrients from our diets. And there's increasing work focused on the possibility that they might even influence neurological functioning. And that body of work, I think, is especially accelerating. But one of the important sort of things to understand is that when you're born, there's a little bit of controversy about this, but essentially you are born, or your gut is born sort of germ-free. They're largely devoid of microbes in your gut when you're born, and then you're colonized. Your gut is colonized by microbes especially rapidly colonized, especially over the first 3 years of your life. They kind of colonize your gut. It's not hard to imagine that the rapid colonization of your gut has an impact, for example, on how your immune system develops. In terms of processing nutrients from your diets, there is definitely, especially within animal models, really great experimental evidence showing how these microbes can basically process nutrients differently depending on which microbes are in your gut. Okay. And there's definitely a lot of interest in this in the medical community, and there's been a load of research in increasing amounts of research in humans, although the body of work in animals is still much larger, and particularly interest in the implications for human health. And many of you out there may have heard, for example, about medical interventions like human fecal transplants to treat something like chronic C. diff infections, which were the most common source of hospital based infections, and for those who end up with chronic C. diff, it's pretty tough to treat. And it turned out these fecal transplants worked pretty well when it came to treating C. diff or chronic C. diff specifically. So there's been a load of research and interest in the implications of fecal transplants and potentially a lot of other interventions in terms of altering human health outcomes. This is just meant to provide just some scale or some sense of some of the work on health. There is evidence around inflammatory bowel disease, irritable bowel syndrome, celiac disease, arthritis, heart disease, C. diff, as I already mentioned, a range of different metabolic syndromes, excuse me, diabetes, obesity, colorectal cancer, and I could put up 1,000 more studies up here that would lay out evidence on the relationship between the gut microbiome and these range of diseases. I would sort of point out one link that is common across many of these diseases as links to, for example, things like inflammation and immune functioning. Those are especially the disease areas where there's, I think, a lot of interest and a lot of evidence about linkages with the gut microbiome. Okay, but why is a social scientist talking to you about this? What is it that why would I be interested as a population scientist or a social scientist? This is a study that came out just about a year ago, actually, that essentially demonstrated that the determinants of the gut microbiome were largely environmental, that the heritability of the gut microbiome taxa is only about 1.9%, and that the overwhelming majority of what shapes the microbiome is a function of environment. Now, I want to be a little bit careful here. My colleague Federico actually does work on how genetic factors influence the gut microbiome, so I don't want to act like this is a closed debate, but I think this is a reasonable summary of kind of where the evidence is about the kind of overwhelming role of the environment in shaping the human gut microbiota. And part of the reason why that's especially interesting as a population health scientist is because the field that I'm in, population health sciences, has essentially decades of research looking at how broader environments, social environments, physical environments, influence morbidity and mortality, and in fact have huge influences on well-being, health, aging, and mortality. And increasingly, that field has gotten interested in the biological mechanisms that can explain, for example, why we see a profound relationship between social relationships and health and mortality. It's one of the best determinants that we know of in terms of health and mortality, and I'll get to an example of that in a minute. But we become more and more interested, basically, in the biological mechanisms or possible biological mechanisms that link those sort of social and environmental factors to downstream morbidity and mortality. And so the possibility that the human gut microbiota might be one of those biological mechanisms, I think, is quite interesting, possibly to many population health scientists. And I do think the population health sciences has some things to offer to the growing, rapidly evolving work on the gut microbiome. So I'm going to be pulling parts of this argument from a paper that came out just earlier this year in Nature Human Behavior with my collaborators, Alberto Piloni, Federico Rey, and Jen Dowd. So one of the things that we can contribute is in terms of thinking about microenvironments. So what do I mean by microenvironments? Well, the existing body of work on the gut microbiome is actually very heavily focused on things like what you eat, individual level behaviors like how much you exercise. A lot of it actually is food and nutrient based. So there's a huge body of work focused on these sort of micro individual level factors that possibly influence the gut microbiome. And indeed, social and population health scientists. These are some examples from the media on some of this work. The New York article on the work around breastfeeding and the gut microbiome, work around nutrition, things like diet soda, exercise. So there's a huge body of work, a lot of which is in animal models, though there is more of this work in humans as well. But these are kind of behaviors in particular that have been a large focus of population health research as well. So a lot of population health research actually has precisely focused on what actually shapes behaviors like obesity, or outcomes of behaviors like obesity or exercise or breastfeeding for that matter, and in turn how those kinds of behaviors influence morbidity and mortality. So this certainly is an area, I think, of overlapping interest between these fields. But in addition to that, I think population health scientists have also spent decades developed huge bodies of research around what I'm going to refer to as sort of macro environments on health and mortality. So these are what we might think of as sort of upstream factors. So for example, social relationships, which I'm going to get into more later in the talk. But one of the, we have decades of evidence demonstrating that very close social relationships are critical in terms of shaping health and mortality. And a lot of that work is actually based on essentially experimental evidence increasingly. Neighborhoods. There's a huge body of work focused on neighborhoods and health and how physical environments can influence health and mortality. That body of work actually also has real experimental evidence. So these large-scale interventions, for example, that effectively put people in different neighborhoods through quasi-or experiments with vouchers, basically, that put some people in some neighborhoods and some essentially randomly assign some people to one type of neighborhood and other people to a different type of neighborhood. That work very much found actually, so they were actually largely interested in looking at influences on things like employment, but what they ended up finding the strongest influences on were on things like health, and particularly on things like inflammation, depression, and stress. And so that's grounded in experimental evidence as well. And finally, pollution. There's this is an area that's grown quite rapidly actually in the past few years, and there's some good quasi-experimental evidence around how air pollution and other kinds of pollution influences morbidity and mortality. So for example, people will do things like focus on populations, comparable populations that live close to highways versus similar populations that don't live quite as close to highways. And you can actually see differences, meaningful differences in morbidity and mortality. It is also the case, sorry, I flipped that slide a bit too fast. It is also the case that those kind of macro environments are of interest to at least a small group of folks studying the gut microbiome. And I'll talk through a little bit of the evidence, but there is evidence actually in each of these domains in terms of social relationships, in terms of neighborhoods, and in terms of pollution about influences on the gut microbiome. That research is really early. A bunch of it is actually, most of it is conducted within the context of animal models, but there is a lot of interest in it. And there's clearly a little bit of empirical evidence around that, specifically around the gut microbiome. So I think there's another area where we have a ton of expertise in how those environments influence health and mortality, and would be real interested to explore the gut microbiome as a possible mediating mechanism, or excuse me, a possible biological mechanism to explain those relationships. In terms of that sort of substantive background, I think we can also contribute in terms of data and sort of general methods. So one of the interesting things going on right now, I think, in terms of gut microbiome research is especially in the past couple of years, the growing number of population-based samples and just generally human studies. So I think for a long time, the work was dominantly in animal models. There has been this growth in human studies to try to understand very specifically the human gut microbiome. And there are some drawbacks, though, to the kind of data that folks are using to do that kind of work. So I'm just going to give you a couple of examples here and talk through a little bit about some of the challenges and sort of limitations, I think, of the existing data. So one example is sort of crowdsourced science, which is really interesting. This is the American Gut Project, where they've collected really large numbers of samples. I believe there's somewhere over 20,000 samples at this point. So people will send in a sample. They pay, I think, around $100, and then they'll also fill out a survey, and then folks have data to use to analyze human samples. Another example, so that's what in the social sciences we would refer to that as kind of a volunteer sample. You're essentially getting people to volunteer. And a large number of the human studies on the gut microbiome at this point are really essentially volunteer studies, like people just sort of volunteering to participate. There has also been a shift, though, towards pushing for more sort of what we would think of as population-based studies. So a really great example of this is the UK Biobank, which is now somewhere around half a million people in the UK. They sent out a broad swath of invitations to try to get people to participate in the biobank. These folks would end up providing sort of a range of different survey data on health and behavior and those sorts of things, that sort of environmental data. And they'd also provide biological samples, including samples like fecal samples for gut microbiome projects. And they ended up with about a 5% response rate, so about 5 for every 100 invitations they sent out. About 5 people participated for every 100 they sent out. The challenge with both the prior project that I mentioned, the purely volunteer sample, and then the somewhat less volunteer-oriented sample, which is the UK Biobank, is that these populations don't tend to be population representative. So they're people who volunteer, and they essentially look a little bit different. So we talk about them as more select. And so there's some key important ways that they're more select. In particular, they tend to be a bit more educated, more well-off, and they also tend to be healthier, importantly. They tend to be healthier. They're less obese, for example. So in this project, in the American Gut Project, I believe somewhere around maybe 10% of that sample is obese, whereas the population-based prevalence of obesity is closer to 40%. This is a study from the American Journal of Epidemiology comparing the UK Biobank to the general population. And in a similar way, it was clear that the UK Biobank sample is healthier, less likely to be obese, so it has that sort of same profile. Now, there's an enormous number of things you can learn from studies like this, even if they're not fully representative of the population. We have loads of examples where we can learn some really, really interesting things. But there's also some real challenges with doing this kind of work on these more select populations in terms of what you're finding. So I wanted to give you a particular example here. So it was really clear from the UK Biobank data. I'm just going to go back here for a second to show you this. If you can see that slide, you can see the UK Biobank. HSC is essentially the fuller population. And you can see the prevalence of most diseases was about twice as high in the regular population as it is in that sample that comprises the UK Biobank. And this is true for essentially obesity as well, or similar kinds of differences for obesity, especially for women. And so I wanted to talk through about the consequences for the kinds of analysis you do when you have these somewhat more healthier select populations in your study. So one of the areas where there has been the most research on the gut microbiome has been around obesity, okay? And one of the findings, especially from animal models, is that higher levels of obesity are strongly correlated with lower levels of diversity in the gut microbiome. So there's less variation in terms of species, basically, in the gut microbiome. And that's generally thought of as not a very good thing. The more diversity on average we think is essentially better for health, or at least that's where the evidence is pushing us. And so certainly from animal models, and then once people started doing more human studies, it really did look like there was some pretty decent evidence that people who were obese compared to lean individuals had lower levels of diversity. Not a great thing. But there was more mixed evidence than I think one might have expected, and there was a recent meta-analysis that came out actually, or sort of an overview of existing findings that, while certainly on average found what we'd expect, which is that people who were obese had lower levels of diversity compared to people who were lean. But the differences, I think, were smaller than maybe we might have expected. So I'm going to talk through about how a select population in your study, in your human study, might cause that, as opposed to it actually being reflective of what's going on in the population. So if you look at these figures, on the left-hand side is what and this is meant to be a sort of stylized example, okay? So we don't actually precisely bin people into high and low levels of diversity. There's variation, but this is just meant to be a stylized example to help talk through the issue. So in the actual population, if you look above, you sort of lean individuals in the population. The yellow groups, we would say, have higher levels of diversity, and then, but you'll definitely have variation. So you have some thin individuals with relatively, say, lower levels of diversity. And then below them, again, this is on the left-hand side, the actual population. Amongst the folks who are, say, obese in that population, you'll see that most of them, say, have lower levels of diversity, but you have variation. So say we have 1 out of 5 who has higher levels of diversity. Now, when we either have a volunteer sample or we have effectively very low response rates, we end up with, as I've already shown you, a healthier population participating in your study. And here's how that can play out. So if you look in the right, in the yellow kind of box there, say the study population can end up looking like this, which is that the folks who are lean, a little bit healthier basically than the actual population, but not a huge difference actually. We can see this in other studies that this can happen when you have a select population. But this is going to happen sort of arguably more to a more extreme extent, essentially, in the folks in the sample who are obese. So the selecting of, you end up selecting people who are healthier on average amongst those who are obese. And so it looks like basically, so say if in the actual population, 1 out of 5 has, in that, higher levels of diversity. Once you have, in the study population, though, it could end up looking something like this, where you have a sort of more 50-50 split. And then the difference between lean individuals and obese individuals looks smaller in the study population than what it would in the actual population. That's precisely how that kind of selective participation can play out in terms of kind of muddying things that you're interested in exploring. The second challenge with the existing body of data and human populations is that they're largely cross-sectional. So most of them at this point are still one point in time, collecting data on one point in time, and don't have sort of, definitely don't have measurements over time on all these sort of broader social and environmental characteristics, where people are living, who they're interacting with, what those broader environments that they live in effectively, what their work environments are like. Those projects, by and large, don't have that kind of very detailed and extensive data. And this is where the social and population health sciences could really be helpful. So I put up a few examples here of NIH-funded studies, or studies that at least to some degree have received NIH funding, including the AdHealth, the Health and Retirement Study, TILDA, which is the Irish Longitudinal Study of Aging, and the National Social Life, Health, and Aging Project, or NSHAP. These are all studies that have been tracking people for at least about 20 years now, and HRS is well over 30 years, has been tracking study populations for over 30 years. You're looking at sample populations that range above 20,000 people. They've been tracked over time. So, for example, in the Health and Retirement Study, they're tracked every other year. And importantly, all of these studies are also collecting increasingly a lot of biological data. So they're collecting blood samples, they're collecting saliva, they're collecting, NSHAP has even done vaginal swabs, hormonal data. They're already collecting this kind of biological data in part to explore these kind of biological mechanisms that might be mediating these sort of broader environments that people live in and health and mortality outcomes. So basically, these are the kinds of projects, and quite bluntly, they have high response rates. They're much, much more generalizable to the populations that they're trying to represent. They're not perfect. They have some selection, but relatively limited selection. Folks spend a lot of time and effort to make sure, in essence, they really limit the amount of selection in these studies. So these are, so essentially, there are data projects, ongoing data projects out there already, these longitudinal studies, for which data on the microbiome could be added, where you could end up exploring some of these questions. So I wanted to give you a specific empirical example of a project that actually did this, which is the Wisconsin Longitudinal Study, the project that I'm the principal investigator on. So this is an empirical example to show you what you can do, basically, if you integrate data on the gut microbiome into one of those sort of traditional behavioral and social science longitudinal studies that already does some biological data collection. This empirical example that I'm going to focus on is going to very specifically focus on the relationship between social relationships and health, and specifically between social relationships and the gut microbiome. Let me back up. I promised I would get to this, and so really one of the most robust findings in the population health sciences is the robust linkages between social relationships and health. And I wanted to give you a sense about the magnitude of those relationships. So what we're looking at here is actually a meta-analysis that was published a few years back that shows sort of the odds of decreased mortality across the range of different predictors, okay, in comparison to social relationships. So we have high social support and social integration, and so you can see both of those, high levels of social support and higher levels of social integration, have significantly increased the odds of mortality, basically, okay? But let's compare that to things like smoking cessation. Actually, the effects, in essence, part of the point of this meta-analysis was to show that effects are certainly comparable, if not potentially larger, for social support and social integration compared to smoking cessation. That's the odds of reduced mortality for drug treatment, for hypertension, physical activity, and being lean versus being obese. And so the point here is just to show you the size of this relationship. So it's a meta-analysis, but there are literally thousands of studies done in a range of different ways, some of which are even in animal models, that really demonstrate the critical nature of social relationships for morbidity and mortality. Now, this literature in the population health sciences, this body of research is huge. And one area in particular that people focus on is marriage. So there's a wealth of data showing that sort of on average, being married, or people who are married are healthier and live longer than people who are not married or who remain single. Now, one really important nuance to this, though, to this body of work, is that it really is a function of relationship quality. So it's not just about your social relationships. It's not just about being married. It's the quality of those relationships that can really matter in terms of predicting morbidity and mortality. And indeed, there's definitely a good deal of evidence at this point that demonstrates this. So you have to, essentially, it's not any marriage that's going to be protective for health. You have to have a good marriage and high levels of relationship quality. Now, interestingly enough, there actually is some work at this point in the gut microbiome that focuses on the possible influences of social relationships. When I first started talking and collaborating with Federico, who's he's a bacteriologist, he mostly works with animal models, and when we first started talking about this, doing this collaboration, and I started talking about, well, here's the big areas of work where we know these are the kinds of social conditions that influence health. And when I mentioned social relationships, he was immediately, that was just so instinctively made sense to him as someone, as a researcher who studies the gut microbiome, in terms of the potential there about how social relationships might actually influence the gut microbiome and even the composition of the gut microbiome, from the kinds of physical interactions that people have in social relationships to their shared physical environments, even stress processes, for example, that might be shared within marriages. So this really instinctively made sense to him given what he knew on the gut microbiome. And when we looked at the research, there is in fact a really interesting and growing body of work in animals. So this is just one example about how social behavior shapes the chimpanzee pan microbiome. And there's some other work here, some other really interesting work as well, essentially finding that these tight social relationships, physical interactions in these primate models does have some influence on their gut microbiome. So we wanted to explore that in the context of a data set that had a really rich data basically on social relationships. If you want to explore in a human model how social relationships influence health, you need a lot of rich data on those social relationships. So we decided to pursue this in this project, the Wisconsin Longitudinal study. I'm just popping up here. This slide shows a few of the different studies that have actually come out, some of which are methodological. The one on top there describes in a lot of detail precisely how the goals of the project, as well as details on how we collected these data. So let me take a step back and talk a little bit about this sample. So the Wisconsin Longitudinal Study is a sample of 1 out of 3 Wisconsin high school graduates from the class of 1957. So they were born approximately 1939. They're currently in their late 70s. The sample now includes actually siblings of many of the graduates, most of the graduates in essence who had a sibling that we could survey. So increasingly it's also a family study. We also collect a lot of data on spouses of the participants. So we've just decided to try a pilot in this project. At the point that we did this, there were no other studies like ours who had attempted to collect data on the gut microbiome and specifically fecal samples. Just to give you a sense about the data itself, we know a ton about these folks over the course of their lives. So we have data spanning from 1957, we're getting back out into the field in 2019. So essentially, actually, from birth until their late 70s, we have data on them. And here's the sort of broader general categories of data that we have: social and family, educational histories, employment histories, job characteristics, a lot of data on children and family, very detailed data on physical and mental health, detailed mortality data, data on things like income and wealth and socioeconomic conditions, detailed data on cognitive performance over most of their life course, including during adolescence. And a lot of this is a mixture of kind of administrative data and prospectively collected data over their lives. We've also added biological data. So we have data on, we have genomic data on well over 9,000 of the participants. And as I already mentioned, we did this pilot study collecting data on the microbiome. So we did this for approximately 427 participants in our study. From our sample frame, the folks we decided that we would go after and try to get these samples from, we ended up with a 76 response rate, which is very high. And we had a mixture of both siblings and spouses as well. So basically fecal samples collected on both sibling pairs and spousal pairs. And we also did a self-administered questionnaire which collected data on things like nutrition and some other kind of critical data that we needed when we collected the fecal sample. I can talk in more detail in question and answer about how we did this, but it's important to understand that we actually went to people's homes to collect these samples from them and to explain to them how to provide the samples. And that was really important in terms of being able to get the kind of response rate that we did and the participation that we got from our participants. So I'm going to briefly walk through here some findings from one of the studies that we did do, and this was now it's published in Scientific Reports. It just came out actually in January, and this shows how close social relationships correlate with the human gut human gut microbiota composition. There's a bunch of different findings from this paper. I'm just going to highlight a few of them. The first thing that we found is that and there was reason to believe this based on prior work, but the prior work was actually less consistent than we initially assumed. So we compared our married couples to sibling pairs to unrelated individuals, and indeed we found that people living together had much more similar microbial composition, shared microbial composition than did unrelated individuals, but also as compared to siblings. So, right, these are folks who grew up together. You know, they spent their whole childhood together, a really important period in terms of establishing, essentially establishing the gut microbiome. But we actually did not find any meaningful differences, any meaningful differences in essence between sibling pairs and unrelated individuals in terms of how similar, the similarity in terms of their gut microbiome. These are unweighted unifract distances that we're measuring. But then it got really interesting because we knew, or I knew, there's this rich social science literature showing, in essence, really big differences between married and unmarried people in terms of health. And I wondered sort of what role the sort of shared microbial composition might have to do with that. So this is real, this is a small study. It has not been replicated yet in another sample, but we have some really interesting findings. So in essence, one of the most interesting findings that we had was that sort of shared microbial composition, so the sort of the degree, the level of similarity in the gut microbiotas of married individuals compared to, for example, unrelated individuals was largely a function of couples who reported very close relationships. So couples who reported that they were just somewhat close really didn't look very different than unrelated individuals. The shared microbial composition of couples, married couples, was largely driven by people who reported that they were very close. Now, this was especially interesting, and it's important to understand this. The married couples in our study, so we did this when they were in their early 70s, even the people who were saying we were somewhat close or have somewhat close relationships compared to very close relationships, these are folks who have been married and living together, in many cases, for well over 40 years. So on the whole, these are actually good marriages, actually. So that sort of difference was really striking to us and really interesting. And then we went a bit further. I think I did already mention this, that in general we think higher levels of diversity and more richness. So this is essentially about greater levels of variation in terms of species in the gut microbiome is important for health, and is a signal at least for better health. And indeed, we did find that married couples, compared to single individuals in our study, had higher levels of diversity and also greater richness, basically. And similar to the prior finding, we found that that was driven by married couples who were reporting very good relationships, or I'm sorry, very close relationships as opposed to somewhat close relationships. So both of those findings were essentially driven by married couples in our sample who had the best marital relationships. And that's super interesting in the context of all of this work, going back 20 years now, that shows how the quality of your marital relationship has an influence on whether or not you sort of get health benefits from that marriage. So I'll wrap up here so that folks have time to ask questions. But in conclusion here, I want to sort of step back in terms of a bigger picture and make a couple of points. One is that the science here is really rapidly evolving. So I want to be real careful here, though, that the science around microbiome is really new or relatively compared to things like research on the human genome. And a lot of that work is conducted in animal studies. There is, as I mentioned, growing amounts of work in human studies. But a lot of the work at this point is correlational rather than causal. And so it's really important to think about that as you're kind of interpreting and considering findings. A bigger picture point, too, is I wanted to just emphasize again that it's not just about social relationships. I gave you, I wanted to walk through a specific empirical example, but there really is some evidence, for example, on how physical environments, neighborhoods, housing, and pollution might influence the gut microbiome. And population health scientists are really well positioned, really very well positioned to help explore those questions. We already have a load of existing data that can be added to help further explore those kinds of questions. So I really do think that sort of tight collaborations between population health scientists and those studying the gut microbiome can be really, have the potential to be really beneficial, both for population health scientists who are interested in understanding possible biological mechanisms that link into our social environments to health and mortality outcomes, but then hopefully also to more basic biologists interested in the gut microbiome and potentially the oral microbiome and other areas of work in terms of the kinds of substantive expertise population health scientists might be able to provide, as well as data as well. So I will stop there, and I look forward to your questions.
Well, thank you, Dr. Hurd. This is Mike Biddle. I'm going to begin the question and answer session. To ask a question or send a comment, click on the Q&A on your screen, select Ask All Panelists, and type in your question and send. So the first question I see here is, in the last study discussed, what were the characteristics that defined a close marital relationship?
That's a great question. So we have a series of questions, basically, where we effectively ask people about how close their relationship is. And then there's a series of categorical responses where they can say whether or not that that relationship is very close, somewhat close, or not close at all. And the married couples in our study basically, again, they have actually on average quite close relationships. So for example, we had maybe we had one couple that reported that they didn't have a close relationship. So we're looking at, differences in marital closeness at the sort of upper end. And we were able to compare actually basically couple level responses in some cases, whether or not there were differences within couples in terms of whether they were reporting. But anyway, yeah, but regardless of how we looked at it, the findings were pretty consistent. So that's effectively how we define it.
Great. So another question that we just received is, were kids of parents shown to have similar gut microbiome?
So there, our work could not speak to that. We don't have the children of these participants in our sample. There is work, so one of the things I didn't talk about is that a lot of the work on kind of families and that sort of shared microbial composition within families is actually done on younger people. There's one exception is really there's an Irish study run out of Cork, but most of this work is actually done on younger people. A lot of this work is done in the developing world. So in those studies, and it's a little bit, you were talking about really different samples when you're talking about Malawi, for example, versus the United States. But anyway, the gist of that work is found absolutely that you see shared relationships within families between parents and children. It's tricky though, because at younger, especially at very early ages, 0 to 3, boy, the evolution there in terms of the colonization of their guts is happening so rapidly. I think some of that work is a little bit difficult, I think, that early on, because things are changing so quickly. But in those studies, they absolutely find evidence of similarity between parents and children.
Another question, can you suggest how junior faculty who are potentially interested in this line of work might explore the field further?
Yeah, well, you can email me. My email is, if you just Google me, you can find my Georgetown email address. So feel free to email me. There is, there's a growing group. So actually, the study that I mentioned, AdHealth, they actually did about a year ago start to collect fecal samples in AdHealth. I don't believe that they've been able to do the metabolomic analysis and things like that yet, but they have data, and so there's a growing group of social scientists doing this kind of work. It's still relatively small, so effectively reach out to me, and depending on where you are and what field you are, I can try to help in terms of linking you up. But I work with actually, or one of the people on our team who I didn't list, but we have some work going on right now with her, is actually at UNC Chapel Hill, and she's a Ph.D. student, so early in her career doing this kind of work. So I'd be happy to talk more basically with you if you email me.
Great. In the last study, were there any identical and or fraternal twins that were included?
So no. So our project only includes, no identical. Our project only includes fraternal twins. So actually data, there's a bunch of work using the UK Biobank, which I already mentioned, where they have really amazing, really interesting work where they do have sort of identical and fraternal twins. The very first study that I put up that showed that the relationship is largely a function, or that the gut microbiome is largely determined by environment rather than genetics, was based on, they employed basically twin and fraternal identical twins to do that. Our data is not. We have siblings basically, but they're not. We don't have identical.
Okay, another question. Do we know about the stability of gut microbiome over the life cycle? For example, 0 to 3 is rapidly evolving. Is that critical period to set the life course?
So that's like the $1 million question as far as I'm concerned. So I'll sort of say sort of generally what people will say, and then I'll talk a little bit about the uncertainty that I think is there. So there's an assumption that there is a certain level of stability. So the way that my collaborator, Federico, will talk about it is kind of like who ends up in there in terms of species is likely to stick with you. What is likely to change over time is sort of things like relative composition. Now, the extent to which they've been able to show that definitively in human samples, though, is really limited. They just don't, they haven't really been able to study populations well enough over the life course, I think, to be really certain about that. You can do it in animal models, and that tells you a lot of really interesting things. But a lot of the human studies tend to compare, for example, there's the sense that there's lower levels of diversity as people get older compared to when people are younger. But those are based on cross-sectional designs, meaning that you're comparing two different cohorts of people. You're mixing between cohort and age, and you can't really be sure if it's actually a function of different cohort experiences, like all the different environments these cohorts experienced, and for some reason older people now look less diverse compared to younger people, or whether it's really a function of something that's going on as people age. So we really need more longitudinal data over time to answer that question effectively, I think.
Your answer to the next question might be interesting. Somebody wrote, has there been any linkage of time use data to microbiome diversity?
Not that I'm aware of, but that would be fascinating. At the moment, we don't have time use data in our project, but that would be fascinating, yeah.
Another question is there a role for machine learning or other data science techniques for associating individual taxa or cohorts of taxa with environmental influences or health outcomes?
Yes. So I do believe, for example, I know that there's some folks at Wisconsin who are really interested in trying to explore, particularly as the data gets more specific in terms of the kind of sequencing they do with samples. I think, yes, I expect we'll see a lot more kind of machine learning approaches to try to help sort some of that stuff out. The one thing I would sort of step back and say, though, is that we always have to be super careful with machine learning approaches. I think that we don't end up with just a wash of sort of associations basically that we get. That you can get with any method, not just machine learning. But I do think because of how, especially once they start doing more, when it becomes more common to be doing more intensive sequencing of these samples, I think they're going to have to start using machine learning to sort some of this stuff out.
So you did mention that this was largely correlational, or the literature, but a question asked is, what are some mechanistic hypotheses linking social relationship to the gut biome? Okay,
yeah. So in my rush to make sure people had questions, so effectively there's a few different ways you can think about it. So one way would be shared nutrition, right? They live together, they're married to each other, they eat the same food. So we actually were able to account for that. And we did not actually see much evidence that we could explain the relationships that we were seeing based purely on kind of shared nutrition. And physical interactions is another mechanism. There is evidence, for example, that that your salivary part, like microbes in your mouth can end up in your gut. And so if you think about, intimate relationships, sharing your toothbrushes, things like that, as well as physical intimacy, that sort of shared intimacy is certainly a possible mechanism. So unfortunately, we weren't able to actually parse out with any certainty what that specific mechanism was, other than to say we're pretty sure that it wasn't just simply about eating the same food. It appears to be more.
Linked to that, a question is, were participants' dietary preference taken into account?
Yes, yeah, exactly. So we essentially collected food diary data. So you go to them. Part of the self-administered questionnaire included food diary data, so that we knew what they were eating. We had questions basically about their general eating patterns, but then also things about what they were especially eating over the days when the sample was provided. So yeah, to the extent to which you can effectively collect that information, we did, and we just didn't see. So for example, we did not see that, well, certainly married people absolutely were more likely to eat similar foods than were people who were related to individuals. We didn't see, for example, that people who reported very close among our married couples, that there was any differences in the commonality of what they were eating across relationship quality. So between, those people who reported very close versus somewhat close, there weren't really differences there in terms of what those folks were eating.
So this is the last question, and they keep coming in. So would you be willing to answer folks who write you individually?
Absolutely, sure, yeah, I'd love to.
Okay, so here's the last question. Can you talk more about breastfeeding and the microbiome? There seems to be disagreement over the importance. What is the cutting edge, biggest data sets, topics being studied?
Wow, it is not. I'll be straightforward here. This is not my area of expertise. I read about it. I'll pick up the articles when they come out. I tend to study later life, but yeah, there's quite a bit of that would indicate that whether or not you're breastfed. Here's what I would say. I think it's pretty clear based on the existing evidence that whether or not you're breastfed influences, composition in your gut microbiome. Sure. What are the long-term implications of that, though? No one has the answers to really say that. There's a lot of to be blunt with you, I hear people overstating what they assume to be the long-term implications without evidence that can fully back up the long-term implications of that. And let me just actually point out, so for example, in our study, we actually went into this thinking that we would see some evidence about sort of a sustained influence of the early life on later gut microbial composition. We didn't see a lot of evidence of that. So, again, like our sibling pairs didn't look any different than unrelated individuals in terms of microbial composition. We did work, for example, looking at whether or not people grew up on farms. So while whether or not you currently lived on a farm had a huge influence on gut microbial diversity and robustness, like that level of diversity of species and stuff, whether or not you grew up on a farm it didn't matter. It had no sustained influence in later life. Now, that isn't to say that differences in early life don't in turn influence things like the way that your immune system develops, but at least in terms of the composition later in life, at least in our project, we're able to find a lot of evidence of sort of sustained influence of early life on later life microbial composition. But it's just one study, and it's a really interesting question.
Great. So this is Christine Hunter. Again, thank you to Mike for moderating the question and answer, and a big thank you to you, Pamela, for your excellent presentation on social and population health science approaches to understanding the human gut microbiome. Also, thank you to all of you who joined online today. If you have colleagues who are unable to join but you think might have an interest, want to remind you that a recording of today's webinar will be available in about 2 weeks on the OBSSR website and on OBSSR's YouTube page. The next webinar will be held on Tuesday, May 14th, and will feature guest presenter Dr. Jeffrey Elgin. He's a professor of medicine and chief of cardiology at the University of California, San Francisco, and he's going to present on the Eureka platform and associated studies. So make sure you're signed up to receive email updates and messages about upcoming OBSSR events. The link to sign up to receive updates is available on our website at obssr.od.nih.gov. And this concludes today's webinar. So we look forward to your participation in May, and thank you for joining, and have a great day.